We already know the definition of cell cycle, and we know that in eukaryotes there are two main events that occur, which is interphase and mitotic stage. For today, we talk about interphase. In simple understanding, we can say that interphase talks about what happens to the daughter cell and what did daughter cell do after the cell division. Thus, we can learn that interphase is known as a preparation stage for the daughter cell. That is, they are preparing themselves before undergoing cell division again. So, in more specific terms, what we're looking at during this phase is that the cell will grow larger in size. Second is that the numbers of organelles increase. And third is that we will see that during this interphase, the DNA will be replicated. So let's see each phase one by one. There are three important phases known as G1, S, and G2. So the first phase is known as G1, or in other words, a recovery stage. Let's say we have a daughter cell after the cell division. You can compare this cell and this cell. What's the difference? So the thing is, the cell will grow larger in size. But why does the cell need to increase its size? Well, because the cell wants to accommodate more space for large numbers of organelles. Indeed, during G1, the numbers of organelles also increase. For example, we can see that our original cell only have one mitochondria. And then from one mitochondria to two mitochondria. So the numbers increase. Other than organelles, they're also accumulating DNA raw materials. What are the DNA raw material? Well, the thing is, we remember that DNA are made up of sugar, pentose sugar, we have phosphate groups, and then we have a nitrogenous base, ATCG. So during this time, the cell will accumulate this DNA raw material. For example, they want to accumulate ATCG, the nitrogenous base. But why? Well, we do know that during interphase, the DNA replication occurs. So, by accumulating large amount of DNA raw materials, for example, like nitrogenous base, ATCG, they are preparing to synthesize DNA in that phase. You might notice as well this yellow circle. So let me zoom in. This yellow circle are an organelle. This organelle is known as centrosome. Inside contains centriole, and you guys might remember that this organelle ate during mitotic stage. Thus, during this G1, from one centrosome is duplicated as well into two centrosome. So, from one centrosome containing one central to two centrosome containing two central. And why this is important? Well, during the mitotic, both of these centrosome will move away from each other, and during that time, they will form a spindle fiber. The spindle fiber will aid during the separation of the sister chromatids or the homologous chromosome during the mitotic stage. Moving on, at S phase, we call this phase as a replication phase. So what happened in this phase? First, the cell will synthesize proteins that are associated with DNA. Now you might wondering, what are the proteins that are synthesized which is related or are associated with DNA? Well, let's zoom in at our DNA strand in our nucleus. Let's say we have a DNA strand from our father. The protein associated with this are known as histone. So what does the histone do? Well, it aids during in how the cell want to organize their DNA. In nucleus, the DNA are actually very spread out, chaotic order, not organized. So the protein histones will allow the DNA strand to wrap around it. So imagine if many DNA wrap around it, they will form a structure. So this structure, if we zoom out, it will look like a chromatin. And in the middle of it, there is in the middle of this chromatin, there is a centromere. Okay, so histones are a protein where the strand of DNA, well, you see this strand of DNA, will coil together this, and then they're gonna form a chromatin. Or we draw it. This is chromatin. Actually, these chromatin are not the typical chromatin that we see during the mitotic stage. The thing is, this is chromatin, but in dense form, it will form chromatin. So, in the middle of this chromatid, or in the center of this chromatid, there is a centromere. Now, back to our phases. We already know that the proteins that are synthesized are associated with DNA, that is, histones. But now, another important point during S phase is the replication of DNA. Since our phase, S phase, talks about replication, what does it replicate? It replicates DNA. So you see from G1SS, notice this chromosome or this chromatid, suddenly they are duplicated. Father only have one, and suddenly there are two. Mother only have one, and suddenly in S phase there are two. During this, we can consider this, for father, we can consider this as one chromosome. In its duplicated form, we can see two DNA are joined together at the centromere. This is also considered as one chromosome. Now, on to the second point is that during S phase is the replication of DNA. One chromatid enter the S phase, and two identical chromatid will exit the phase. So you can see that, notice there is only one chromatid. When it enters the S phase and exits out the S phase, it will form two identical chromatid. These identical chromatids are known as sister chromatids. Both of the chromatids have a centromer. For example, this and this both have a centromer, and they are joined together at the centromer. During mitosis, these sister chromatids will be separated at the centromere. Now, the third phase will be a G2 phase, known as a preparation stage. Now, why does we call this as a preparation stage? Because during this time, the cell actually synthesize proteins that will aid in cell division. So, what is the protein that are synthesized that aid in the cell division? We already know that during G2, there are protein associated with DNA such as histone. What's the difference between G2 and S phase? Well, the thing is, the protein that the cell synthesized during G2 are different than the S phase. The proteins that are used to synthesize in S phase are used to organize the DNA in a coil form. But in G2, this protein actually will form a microtubules. Microtubules will form spindle fibers or mitotic spindles. So in S phase, the proteins are histones that are used to organize the DNA. Meanwhile, in G2, the proteins are used to synthesize protein that will form microtubules, and these microtubules will form spindle fibers. So these are the proteins that form microtubule. You can notice there is a microtubule near the centrosome. So what happened during the mitotic stage? The centrosome will use this protein to build microtubules that will form a spindle fibers that will attach to the chromosome and allow it the division of the chromosome at its centromere. So that's all for the interphase.